Hello everyone, I am K. Devavani, Assistant Professor of Microbiology from Government Degree College for Women, Nalgonda. In this video, I am going to explain on antigens. I explained the antigens in the previous video, that is about the introduction, types of antigens, chemical nature of antigens, properties of antigens. In this video, I am going to explain about the specificity of antigens, that is antigenic specificity, and the biological classes of antigens. Let me briefly go through about the contents of what are antigens and what is the chemical nature of antigens and the properties. Just summarizing the previous video, that the antigens are the substances that induce the immune response when enters into the host and react with the products of that immune response. There are two terms, immunogens and antigens. Immunogens are the substances which can elicit the immune response, but the antigens are those substances which can react with the products of that immune response. Based on the immune response, antigens are two types, complete and incomplete antigens. Complete antigens are able to induce the immune response and they can react with the products of the immune response, that is the antibodies and the Tcell receptors. Incomplete antigens are those antigens, they do not induce the immune response, but they are able to react with the products of immune response. They are also called as haptens. Haptens here, incomplete antigens, can be converted into complete antigens by means of carrier molecules. Again, they are simple and complex. Epitope is the smallest area of the antigen that can bind to the antibody. Epitopes are also called antigenic determinants. Epitopes are of two types, linear epitopes and conformational epitopes. T cells recognize linear epitopes and B cells recognize conformational epitopes. Some of the factors that influence the antigenicity include the molecular size, chemical nature, physical form, foreignness, degradability, dose and route of the antigen, and adjuvants. So, in this video I am going to explain about the antigenic specificity. What is the meaning for specificity? Here the antigens, as explained in the definition, they react specifically with the products of the immune response. They stimulate the immune response and it reacts with the products of the immune response. The products of the immune response are the antibodies or the T cell receptors that are present on the T cells. Specificity is an important feature of all the immunological reactions, and the specificity is related to stereochemical nature. That stereochemical nature means here the arrangement of the molecules, position of molecules or the arrangement of molecules in space. Even the stereo configuration, it determines the antigenicity. The stereochemical specificity was first demonstrated by Obermeyer and Pick, and it was later confirmed by Landsteiner. The stereochemical specificity was demonstrated by Obermeyer and Pick, and it is confirmed by Landsteiner. Spatial configuration influence the antigenic determinant and the antigenic specificity. Antigenic specificity is not absolute here. The cross reactions occur in between the antigens. The cross reactions occur because of the stereochemical similarities that are present in between the antigens, and also sometimes the cross reaction is due to the sharing of the identical antigenic determinants. Antigenic determinants are also called as epitopes. So, the cross reactions also occur due to the sharing of epitopes by the different antigens. The specificity of tissue antigens of animals is considered under these categories such as species specificity, isospecificity, autospecificity, organ specificity and heterophile specificity or heterogenetic specificity. So, first let me explain about the species specificity. Species specificity, it refers to the species means tissues in all the individuals of a species contain these species specific antigens. Tissues in all the individuals, all the members of a species contain species specific antigens. Species specific antigens, generally it shows the phylogenetic relationship. This phylogenetic relationship, they are useful to trace the evolutionary relationship between the species, and also these species specific antigens are helpful to identify the species in forensics applications. In forensic applications, from the blood and seminal stains, these species specific antigens are used to identify the species. Next one is isospecificity. Isospecificity, it refers to the iso antigens, means that these are the antigens present in some of the members of a species but not in all the members of a species. So, the species here are grouped based on the presence of different iso antigens in the members. The best examples for these iso antigens are the blood group antigens. They are human erythrocyte antigens. Blood group means that is a erythrocyte antigens with human erythrocyte antigens, and another one is a histocompatibility antigen. Blood group antigens are genetically determined, and blood group antigens have a importance in blood transfusion and isoimmunization during pregnancy, and these blood group antigens are also helpful in determining the paternity cases, but of course nowadays these are supplanted by the DNA fingerprinting tests. Histocompatibility antigens are the antigens which are the cell determinants and specific to each individual of a species. They are specific to each individual of a species and they are the cell determinants. The best example, HLA. HLA is a major histocompatibility antigen that determines the homograft rejection. Human leukocyte antigen, HLA typing, it is called as a human leukocyte antigen. It is one of the major histocompatibility antigen that determines the homograft's rejection. HLA typing is essential before carrying out the transplantation of tissue or the organ from one person to another person. When the transplantation is to be carried out from donor to a recipient, HLA typing is essential before the transplantation process. Next one is autospecificity. Autospecificity means, here auto means self. These are the self antigens. They are not antigenic, but there are exceptions. Exceptions are seen in the case of I-lens protein and sperm, because I-lens protein, here it is a sequestrated, means it is a hidden from the cells of the immune system. So, these sequestrated antigens, they are not free in circulation or the tissue fluids, they are present within its capsules. So, they are not recognized as a self antigen. Same way, the sperm, it is not present during the embryonic life, which is developed later in the life. So, it is generally not recognized as a self antigens, though they are the self proteins, but they are not recognized as a self antigens. Next one is organ specificity. Organ specificity refers to the organ specific antigens. Organ specific antigens, they are confined to particular organ. Antigens, they are characteristic of an organ or the tissue present in different species. Here examples for such organ specific antigens or the proteins include the proteins that are present in the brain, kidney, thyroglobulin and lens protein. The proteins of brain, kidney, thyroglobulin and lens protein of one species share the specificity with the other species. The antigens which are characteristic of an organ or the tissues, they are present in different species, are called as the organ specific antigens. Examples for such organ specific antigens include the complications. The complications are observed after the anti-rabic vaccination using the sheep brain vaccines. Vaccines, here the anti-rabic vaccines are prepared using the sheep brain. When these vaccines are given to the individuals, the complications, neuroparalytic complications will be observed. It is due to the brain specific antigens shared by the sheep and a human being. Human beings and sheep brains, they share the organ specific antigens that leads to the neuroparalytic complications after the anti-rabic vaccination. And the next one is heterogenetic or the heterophile specificity. Same antigen or the closely related antigens sometimes occurs in the different biological species, classes and kingdoms. The same antigen or closely related antigen present in the different biological species, classes and kingdoms. Such kind of antigens are called as a heterophile antigens or heterogenetic antigens. Examples is a Forceman antigen. Forceman antigen is a lipid carbohydrate complex It is a lipid carbohydrate complex present in the animals, birds. It is same antigen is distributed in the species, classes and the kingdoms. So, animals, birds, plants and bacteria. The Forceman antigen is an example for heterophile antigen, which is a lipid carbohydrate complex that is widely distributed in the animals, birds, plants and bacteria. It is absent in rabbits. So, to raise the antibodies to the Forceman antigen, that is anti-Forceman antibodies are prepared in the rabbits. There are other heterophile antigens which have importance in the diagnosis. Such as a Weil-Felix reaction, used for diagnosis of typhus fever and cold agglutinin test in primary atypical pneumonia. The specificity of tissue antigens of animals are considered to be either species isospecificity, autospecificity, organ specificity and heterophile specificity. Species specificity is the tissues in all the members of the species. They contain the species specific antigens that leads to the cross reactions because the antigens are from the related species. These species specific antigens are useful to trace the evolutionary relationship and they have a forensic application in identification of species. Isospecificity is the best example here is isoantigens and histocompatibility antigens. Isoantigens are the human erythrocyte antigens based on which the individuals are classified into different blood groups. Histocompatibility antigens, they have a major function in the transplantation. So, HLA human leukocyte antigen here is a one of the major histocompatibility antigens that determines the homograft rejection. Autospecificity, they are the self antigens, generally they are non-antigenic, but some exceptions are seen such as in the lens protein which is confined to its capsule, but it is not found in circulation, but it is antigenic. And another one is a sperm which is not produced at the embryonic life, which will be developed later, will serve as also an antigenic. Organ specificity, it is organ specific antigens here confined to a particular organs. Examples are the antigens that are present in the brain, kidney, thyroglobulin and lens protein of one species. They share the specificity with the other species. Another one is the heterophile specificity. Same antigens or the closely related antigens are present or they are distributed in the different biological species, classes and kingdoms. Example is a Forceman antigen, which is a lipid carbohydrate complex that is distributed in the animals, birds, plants and biological classes of antigens. Here, depending upon the ability to induce the antibody formation, the antigens are classified into T cell dependent antigens. TD means T cell dependent antigens. Another one is the T cell independent antigens. They are called as TI antigens. T cell dependent antigens, these are the antigens, they require the involvement of T cells. Most of the naturally occurring proteins are the T cell dependent antigens. T cell dependent antigens are captured by the antigen processing cells, or which includes macrophages, dendritic cells and B cells, or these T cell dependent antigens, they can also directly bind to the immunoglobulin receptor that is present on the B cells. And now these antigens are processed, they are internalized by means of a phagocytosis and they are processed into smaller peptides, which are then expressed on its surface complexed with MHC2. Once this complex, that complex here it is a tertiary complex. The tertiary complex includes the T cell receptor that is present on the T cells and antigenic fragment and which is complexed with the MHC2. So, it forms the antigenic interaction forms a tertiary complex. It includes the TCR, antigen and major histocompatibility complex 2. Now, the T cells they bind to this antigenic peptide which is complexed with MHC2 and gets activated. That activated TH cells now release cytokines and that cytokines again activate the B cells. These B cells now produce the antibodies. T cell dependent antigens, they require the involvement of T cells. The T cell independent antigens, there is no involvement of T cells. Some antigens, they directly stimulate the antibody production by B cells without the participation of T cells. Such antigens are called as the T cell independent antigens, the antigens which can stimulate the antibody production directly by B cells without the participation of T cells. Such antigens are called as TI antigens. These antigens, they react with the BCR, B cell receptor. Based on this, the antigens are again T cell independent antigens are two types, type 1 antigens and type 2 antigens. Type 1 antigens include endotoxins. They are directly mitogenic. Mitogenic means they induce the mitosis of B cells. Division, the B cells undergo division. And the type 2 antigens, they are the polysaccharides. The polysaccharides, which are present in the bacterial cell wall. The type 2 T cell independent antigens include the bacterial cell wall polysaccharides and the cap polysaccharides, and also some proteins like flagellar proteins. Bacterial cell walls and capsular polysaccharide and the flagellar proteins, they serve as the type 2 T cell independent antigens. These type 1 and type 2, they activate the B cells and produce antibodies and the cytokines, and they also activate the other cells like macrophages, dendritic cells, mast cells, and also activate the complement. So, the T cell independent antigens, they activate the complement, but activation is not observed in the T cell dependent antigens. Isotype switching occurs. Here isotype switching means the first immunoglobulins are IgM, earlier immunoglobulins, and later specific antibodies specific to the epitope will be produced. But in the T cell independent antigens, it is the isotype switching is limited. In the T cell dependent antigens, immunological memory is present, but no such memory is observed in this T cell independent antigens. Here in the T cell dependent antigens, all classes of antibody like IgM, IgG, IgA, IgE, all classes of antibody will be produced, but in the Ti antigens, it is the earliest antibody IgM and later IgG3 will be produced. And the next one is they are easily degradable, because present to the T cells, the antigen should be processed, it should be phagocytosed. So, they should be easily digestible or degradable to the tissue enzymes, but here there is no requirement of T cells, they are poorly degradable. The T cell dependent antigens are mostly the proteins, but here when it comes to Ti antigens, they are the, as explained, they are type 1 and type 2, they are endotoxins and lipopolysaccharides and the polymeric proteins and the polysaccharides that are present in the bacterial cell wall or in the capsule. So, these are the classes of antigens based on the formation of antibodies. Ability to induce the antibody formation, and antigens are classified into two types, T cell dependent and T cell independent antigens. These are the differences. Next, move on to the super antigens. Antigens are nothing but a proteins, molecules, but these they activate the large number of T cells irrespective of the antigenic specificity, and they secrete a variety of the activated T cells, again they release cytokines. These have a profound influence on the immune system. A more number of cytokines will be released by these activated T cells. Large number of T cells are activated by these super antigens. Super antigens are the protein molecules that activate the large number of T cells irrespective of the antigenic specificity. In the conventional antigen presentation, as explained in the previous videos in the CMI, the antigen presenting cells, which is called as APC. APCs are three types, they are macrophages, dendritic cells and B cells. Once the antigen is processed into antigenic peptides, the MHC2 is present on the surface of APC cells. So, this is an antigenic peptide. This is an antigenic peptide which should be complexed with this MHC2, and these are expressed outside on the surface of the antigen presenting cells. In the conventional method of antigen recognition by the T cells, the T cells have the TCR. This is called as a T cell receptor that is present on the surface of the T cells. It contains alpha and beta fragments. This is the groove that is produced by the T cell receptors that fits the groove that contains the antigenic peptide. This is the conventional way of antigen presentation to the T cells. But in case of super antigens, what happens? This is a T cell which is having the T cell receptor with alpha and beta subunits, and antigen presenting cells, antigen is processed and it is complexed with the MSC2, but the super antigen here not it is in the groove that is formed by this complex, but it binds to the lateral part of the groove. It is not in the alpha beta dimer groove of the MSC molecules, but it is in the V region, that is the variable region, and this region of the TCR which contains alpha and B chains. Super antigens, they bind to the lateral part of this groove, but not within the groove that is formed. It is irrespective of the antigen specificity. These super antigens, they bind to the lateral part of the groove that is formed by the alpha and beta chains. The super antigens are characterized by the resistance property. They are highly resistant to the proteases enzymes, and they are resistant to the denature by CD4 T cells. So, this leads to the release of cytokines. Huge number of cytokines will be released, one of which is the IL-2. This IL-2, it causes the massive proliferation of T cells. Again, this IL-2 causes the massive proliferation of T cells. It leads to the further release of cytokines. So, these have a effect on the immune system. The super antigens are two types: exogenous super antigens and endogenous super antigens. Exogenous super antigens means these are the bacterial super antigens. Examples of such kind: Staphylococcus aureus enterotoxin. Staphylococcus aureus enterotoxin, it acts like a super antigen. This enterotoxin, it causes the disease of food poisoning or the toxic shock syndrome. Another one is a streptococci, which release exotoxins. These exotoxins also act like a super antigens. It also causes a shock, septic shock and rheumatic fever disease. Another one is endogenous super antigens. The endogenous super antigens, they are AES, AGs. They are the cell membrane proteins. They are coded by the viruses. The viruses that are infecting the mammalian cells, they have the proteins on its surface. Particularly viruses, enveloped viruses, they have the glycoproteins on their surface. In case of HIV, where ESAG is coded by a ENV. ENV means envelope gene of a human endogenous retrovirus. These are the super antigens. Endogenous super antigens are coded by the viruses that are infecting the mammalian cells. So, this is an antigen presenting cells as an alpha and beta subunit to peptide, but that peptide is not specific to the TCR. The peptide is not specific to the TCR. TCR again contains alpha and beta and constant and variable domains. This is the variable domain, and super antigen here, the endogenous super antigen which is bound to the membrane, that is the antigen presenting cell. This not in the groove, but it is in the lateral part, sides, that to the side of this alpha and beta domain of TCR, this super antigen binds. That leads to the massive proliferation of and activation of T cells and the cytokines. This T cell activation and the proliferation. So, the super antigens are the proteins that binds to the TCR variable beta chain and alpha chain of MSC class 2. MSC class 2 molecules are present on the antigen presenting cells. This is an MSC class 2 molecule to which the antigen should be bound, but here the peptide is present which is not specific to the T cell receptor, and the super antigen is bound, it is onto the lateral part. So, that leads to the massive activation of T cells and release of large number of cytokines which have the physical effects. So, with this the topic on the antigens. In this antigens, what is to know here means what is an antigen and what is an hapten, and what are the epitopes, what are the different factors that influence the antigenicity, the specificity, what are the isoantigens, and the adjuvants. What are adjuvants? These are the substances that increase the immunogenicity. Example for such adjuvants include alum, friends, incomplete adjuvant, friends, complement which contains inactivated tubercle bacilli, and the super antigens. The super antigens again exogenous and endogenous. Exogenous bacterial and endogenous is viral. The bacterial example is staphylococcal enterotoxin and streptococci exotoxin. Endogenous, the examples include HIV and another examples like rabies virus, Epstein-Barr virus, etc. With this, the topic antigens is completed. Thank you for watching this video.